Welcome, everyone, and thank you, Taka and the Holland Biomedical Clinic, for hosting this event. Today we're going to talk about mending mitochondria with microcurrent therapy. Before we dive into microcurrent, I think we're going to do a little review on mitochondria and why they're important. Mitochondria reside within the cell. You can see them in the sort of the reddish pink oval shape within the cell, and their sole purpose is to create energy. And energy comes in the form of something called ATP. Think of ATP as your cash or your currency used by every tissue, every cell in the body to generate or do their work. 90% of the oxygen that we inhale is used to generate this ATP, and the food that we eat is broken down to create, ultimately, ATP. So our food is used as the fuel for the mitochondria to create the ATP energy. When food comes into the body, such as carbohydrates, fats, and protein, they go through individual processes and are broken down so that they can enter the mitochondria and the Krebs cycle to generate ATP. Mitochondria reside within the cell, and you can see them in the sort of the reddish pink oval shape within the cell, and their sole purpose is to create energy, and energy comes in the form of something called ATP. Think of ATP as your cash or your currency used by every tissue, every cell in the body to generate or do their work. 90% of the oxygen that we inhale is used to generate this ATP, and the food that we eat is broken down to create, ultimately, ATP. So our food is used as the fuel for the mitochondria to create the ATP energy. When food comes into the body, such as carbohydrates, fats, and protein, they go through individual processes and are broken down so that they can enter the mitochondria and the Krebs cycle to generate ATP. So in this slide, let's just review. There are basically three phases to the production of ATP or energy. The first phase is the consumption of food and the breaking down of that food, fats, carbohydrates, and proteins into smaller substances like acetyl-CoA, pyruvate, and amino acids, so that those things can then be shuttled into the mitochondria to help drive the next phase, which is the citric acid cycle. The citric acid cycle, phase 2 of energy production, requires additional cofactors to actually run. Think of it as a cycle going round and round, but it really needs not just the fats, carbohydrates, and proteins. It needs additional vitamins, minerals, and coenzymes to work, and some of those are magnesium, which is critical throughout the body in a number of enzymes. But niacin, also known as vitamin B3, is also very important in the Krebs cycle. Niacin makes something called NAD+. NAD+ then attaches to a hydrogen electron, also sometimes known as a proton, and shuttles that into the third phase, known as the electron transport chain. So the NAD+ is the electron carrier, or the hydrogen ion electron carrier. Riboflavin, another B vitamin, vitamin B2, creates something called FAD+, and that FAD+ then attaches itself to another hydrogen ion and then transports that into the electron transport chain. These NAD+ and FAD+, if you look at the slide on the left at the bottom, they're really the products of that entire citric acid cycle. The whole thing goes round and round, but it really is going round and round to produce these electron transport carriers, NADH and FADH2, that will then move into the third phase, which is the electron transport chain. CoQ10, also known as ubiquitol, we'll talk later about how important that is in the electron transport chain, the third phase. You should also know carnitine is an important coenzyme, cofactor. It's used to shuttle fats into the mitochondria. And ribose is something else that's important to the production of ATP energy. It's used to replenish ADP. So you can see that Krebs cycle is a really important part of energy production by the mitochondria ultimately to make ATP. And so I thought we might review that the Krebs cycle. And I found a YouTube video online that's done by a high school biology teacher, and he uses rap to review the Krebs cycle. Now, mind you, he's not the best rap singer, but he's a very good teacher.
Matrix of the mitochondria of all our cells is the cycle of reactions that one hands Krebs a Nobel Prize. The cycle takes the energy in food and makes it into other forms your cells can use. Krebs cycle makes electron carrier NADH, which later brings electrons to the electron transport chain. And Krebs makes FADH2, its function is the same. Krebs also makes some ATP, another claim to fame, we're talking Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes one ATP, 3 NADH, 1 FADH2. Right before the cycle's a transitional part, links Krebs to glycolysis so Krebs can start. Enzymes break a CO2 off a pyruvate, yields an NADH and acetyl-CoA. Acetyl-CoA carries carbons too, with all the energy that derives from food. It's a highly reduced molecule with energized electrons that fuels up the Krebs cycle as it cycles on. Now at the start of Krebs, this acetyl-CoA has the 2 carbons it carries in, ripped away. Enzymes put these on oxaloacetate with carbons 4, makes 6 carbon citric acid. Who could ask for more? Notice 3 carboxyl groups on the citric acid. That's why the cycle's also named for tricarboxylic acid, or TCA cycle if it's acronyms that you prefer. Krebs in honor of its discoverer, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2. Enzymes work on citric acid and remove the CO2, and other enzymes modify and oxidize it too. The 5-carbon result is alpha-ketoglutarate. Krebs cycle, it's so great. Every oxidation can power the reduction of NAD+, which gains electron carrying function, become an NADH, that energy sensation, which later on in respiration powers ATP creation. Another oxidation follows another CO2 remove, leaving us with a 4-carbon molecule. Another NADH results from this oxidation. As Krebs does its energy transformations, this 4-carbon molecule, succinyl-CoA, still has lots of energy enzymes can take away. A series of reactions yield 1 ATP, the cell's main energy currency, leaving just enough energy for enzymes to reduce NFADH to FADH2, and 1 last NAD+ will also get reduced as the final electron carrier NADH gets produced. We've harvested what energy came in at Krebs to start. Now we have oxaloacetate at this final part. Oxaloacetate is the commencement and finale, ready to meet acetyl-CoA, and here at the final tally. Krebs goes round and round, such an ancient cycle, spinning like the wheels of my bicycle. Krebs is like the axle of aerobic respiration. I breathe out of CO2 with every exhalation. We've walked through the cycle, so now let's review. Input is acetyl-CoA with carbons too. The carbons get removed, releasing CO2. Exhaling sends the CO2 out of you. The cycle's functions energy transformation, 3 NADH, 1 FADH2 creation, and also synthesis of 1 ATP, which cells directly utilize for energy. For every glucose cells absorbed, the cycle runs 2 times. As long as cells get fuel, Krebs is running just fine. It precedes electron transport chain, it follows glycolysis, it's spinning round and round in the mitochondrial matrix, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2.
If you enjoyed that last YouTube video, you can go to his Mr. W's YouTube channel and find other videos that teach biology. It's pretty clever. So just reviewing again the Krebs cycle. The Krebs cycle creates 3 NADH hydrogen ion carriers, hydrogen electron carriers, 1 FADH2 and 1 ATP. Carnitine shuttles long chain fatty acids into the mitochondria, which is why we need carnitine. And then the short chain fatty acids and medium chain fatty acids can enter on their own without carnitine. Fatty acids are broken down to create acetyl-CoA that then feeds into the Krebs cycle, and the carbohydrates are broken down to pyruvate that feeds into the Krebs cycle. So if you look on your left, you see again the bottom of the slide, food molecules are broken down, fatty acids, pyruvate, enter the cell, go into the Krebs cycle, NADH, FADH are produced, and now the third phase is the electron transport chain at the top in yellow, also called oxidative phosphorylation, that moves those electrons down the chain and ultimately help to create ATP. So just to review electron transport chains, energy production, as you can see above, you get 1 ATP from the Krebs cycle, but from the electron transport chain, you get 28 ATP. So that's really where the energy is made. And again, you need niacin and riboflavin and CoQ10 and ribose all in the electron transport chain to help generate that ATP. The next slide is another video. This one's quite elegant, an animation of the electron transport chain and how that works.
All eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the four protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the four complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits two high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal two reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in two important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
So now that you understand how mitochondria work and produce energy, let's look at how mitochondria become deficient or inactivated. So the first obvious thing that can make the mitochondria dysfunction are deficiencies. So we talked about how important niacin and riboflavin, vitamin B3 and vitamin B2 are in creating NAD plus and FAD plus that create the electron carriers that travel to the electron transport chain. So if you're deficient in B vitamins B3 and B2, that can affect your mitochondria. If you're deficient in CoQ10, which works in the electron transport chain to shuttle those electrons across the electron transport chain, that can also affect mitochondrial function. Magnesium, which is needed everywhere, low magnesium. Ribose becomes depleted when ADP is depleted. If you have a lot of inflammation in the body and you're using up all of your ATP, you have to revert to ADP for energy, and so then that ADP supply gets used up. It's not as efficient as ATP, but it gets converted into ATP. So when you deplete your ADP, then you're really affecting mitochondrial function. Carnitine, if you're deficient in carnitine, you can't shuttle those fatty acids into the mitochondria, and fats make so much more ATP than carbohydrates, so it's important to have enough fat for the mitochondria. If you're deficient in antioxidants like vitamin C, vitamin E, then the polyphenols that come from fruits and vegetables, the oxidative stress, which is kind of the byproduct of the energy that's made, you create sparks, if you will, that can actually damage nearby tissue. Antioxidants come along and neutralize the oxidative stress. So if you don't have enough antioxidants around, you're going to harm your tissue, and your mitochondria become less efficient. And many of the things above list are included in something called the mitococktail. Those are nutrient supplementations to help support mitochondrial energy. Another whole category that can affect mitochondrial efficiency are the B vitamins, B vitamin deficiencies, and then methylation SNPs. It's a different pathway than the Krebs cycle and what goes on in the mitochondria pathway, so it's like an entire lecture to itself. But B vitamin deficiencies in general, or conversion of B vitamins to active B vitamins, can also affect mitochondrial function. Another cause of mitochondrial dysfunction is the accumulation of toxins or cell waste products, and those can include heavy metals such as arsenic and lead and mercury and aluminum. You can see to the left there are different places along the Krebs cycle, or the conversion of fats and carbohydrates and proteins, that heavy metals can affect and interfere with that pathway. Pesticides and environmental chemicals also can affect mitochondrial function. The mitochondria are very sensitive to some of these chemicals. If you're not getting enough oxygen to the tissue, or you don't have enough of the other substrates, cell waste products can accumulate and also affect the performance of mitochondria. And then again, lack of sufficient oxygen or even perfusion to that tissue can affect the performance of mitochondria. Recent research has uncovered another cause of mitochondrial dysfunction in children with autism, and that's dysbiosis. In particular, it's the overgrowth of certain bacteria, like the Clostridia species, that appear to produce excess propionic acid. Propionic acid is a normal short-chain fatty acid produced and used by our body. Just to review, if you look at the slide on the left, it's the Krebs cycle, and then up on the right, in the right-hand corner, is the electron transport chain. So you see the right side of the Krebs cycle is producing 2 NADH, and the left side of the Krebs cycle is producing 1 NADH and 1 FADH2. And these are electron transport carriers, then take the electrons and dump them into the electron transport chain to ultimately produce ATP. On the right side of this slide, in the upper right-hand corner, you see when there's overgrowth of Clostridia and excess propionic acid is produced, that propionic acid jumps ahead of the left side of the Krebs cycle and enters at the bottom in succinyl-CoA. And then in doing so, it basically shuts off the entire, as you look at the slide to the right, that entire right side of the Krebs cycle. And the result of this is a decreased production in NADH. So you lose the 2 NADH produced on the right side, and you only produce 1 NADH and 1 FADH2. So you have about a 50% reduction in hydrogen ions that get dumped into the electron transport chain, which in turn dramatically decreases the production of ATP. If excess propionic acid is causing dysfunction of the mitochondria in the Krebs cycle, you will find clues on the urine organic test. What you'll see is as the citric acid cycle goes round and round, the byproducts or organic acids that aren't used are then dumped into the urine. So if you're not using the right side of the cycle, the citrate, isocitrate, alpha-ketoglutarate, you're going to see some increase in those organic acids in the urine. And then on the left side, when you're dumping more of the propionic acid into the cycle, you'll see an increase in 3-nitropropionic acid in the urine. So you get clues from that urine organic acid test about how well that citric acid cycle is functioning. Another major cause of mitochondrial dysfunction is inflammation. And we've had a number of devastating hurricanes recently, so you might even think about inflammation, uncontrolled chronic inflammation in the body as a hurricane. You know, it does a lot of damage, and it essentially creates a lot of debris, can damage infrastructure and cellular membranes, causing an overload of debris in your lymphatic system, in your bloodstream, in your tissues. So it really looks a lot like a hurricane. Chronic inflammation is toxic to mitochondria. And there is something called NF-kappa beta, which acts like the eye of the storm. NF-kappa beta is what causes a cascade of downstream inflammatory mediators. So when that NF-kappa beta gets turned on and doesn't get turned off, you continue to see like a hurricane effect. You get widespread cell and tissue destruction. The cells are overwhelmed and can't clear the debris and waste products. You get depletion of nutrients and cofactors because you're overusing your mitochondrial energy supply. You see retention of fluid and swelling in tissue, and then you get slow movement of nutrients both into the cell and slow movement of toxins out of the cell. So just to review, the main source of energy production is hydrogen ions that go into the electron transport chain and generate ATP. Dysfunction of the mitochondria can occur as a result of nutrient or cofactor deficiencies, toxin accumulation, low oxygen, and inflammation. So let's switch gears now and talk about microcurrent therapy. So microcurrent is an extremely low-level electrical current that is used essentially by our cells. Our cells all have an electrical charge in them, and microcurrent replicates that low-level electrical current. And then within the tissues in our cells and our bodies, there is a frequency in addition to the level of the current that are unique to that tissue and some of those cells, and that keeps them functioning. So how does microcurrent work? Well, it's been found that microcurrent results in a 500%, on average, 500% increase in ATP production. It's also been found to reduce NF-kappa beta, and therefore it reduces inflammation. And it also is found to increase the electrons production, and that can then be used for energy production by the Krebs cycle and electron transport chain. So the result of microcurrent therapy, or the use of microcurrent through the skin and down into the tissue, is you get a reduction in inflammation, you get improved mitochondrial energy production, you get improved circulation and cell function, you get more rapid tissue healing and repair. And another advantage is that it can actually have an effect on brain waves, and you can get a more healthy reorganization of brain waves. There are 5 different types of brain waves, and in general, we function in either delta, which is a deep sleep, or delta theta, which is kind of a drowsy sleeping, kind of a meditative autopilot, or an alpha state, which is more of a relaxed state and still functioning, and then beta, which can be the more alert, even high beta, which would be almost hyper alert, anxious. So at any point in time during the day, we're in one of these states. Normally, people spend most of their time in the alpha frequencies, but when researchers studied children with autism, they found that this curve was flipped to a U-shaped curve, as you can see on the left, that children with autism spend less time in the alpha, that relaxed state, and have more time in the high beta, which is the anxious state, or in the delta, kind of low delta, which is kind of in that zone, if you will, that almost sleepy, inattentive zone, which might explain correlate with some of the behaviors like inattention, anxiety, and some of the social behaviors of autism. So microcurrent therapy can also be used to modulate some of the brain waves, and we can use some of the lower, slower frequencies to take kids from the high beta down to a lower frequency in a more relaxed state. Or we can take kids who are way too relaxed and inattentive and try to get them in more of an alpha beta state so that they can pay attention and focus more. So in summary, microcurrent can be used for a variety of conditions, and its effects are thought to accrue over time with treatment. There isn't one and done. Generally, it requires a series of treatments, but it usually can be sustained once you achieve that repair and healing. Microcurrent therapy is generally safe. They're FDA-approved devices, and they're not known to have significant adverse effects. It's not recommended, however, to be done on patients who are pregnant or who have implanted pacemakers, because the effects are just not known. In our clinic, we generally are using microcurrent for anxiety, insomnia, and depression, for neuropathies and strokes and sports injuries. Although it can be used for a variety of conditions, as you see listed on the right. The instruments that we use in our clinic are the electroacuoscope on the left, and then beneath it on the right is the electromyopulse. And these instruments use an alternating frequency and have computers that can actually read impedance within the tissue, so they deliver a specific current to tissues where it's needed. There are a variety of other instruments, though, that can be used to deliver microcurrent. Some of you may have heard of cranial electrical stim devices that are more of a pocket-sized mobile device that can be used as ear clips and to deliver like an alpha wavelength and help to reduce anxiety, insomnia, and depression. There's some nice studies showing that stimulation with alpha stim compared to drugs, such as on the right, under anxiety, you see Xanax and Ativan compared to alpha stim, alpha stim had as much of an effect as the drugs, and perhaps even a better effect. And the same was true for insomnia and depression. So these devices are really quite handy for individuals for home use that are struggling with anxiety and insomnia or depression. Here are a couple of examples of how microcurrent is used in our clinic. We do have a headband device that can just wrap around the head, and then we deliver the frequencies through the headband device. It's quite beneficial for those with anxiety, or sometimes as a pre-treatment before we move in to treat in other areas such as inflammation in the abdomen. And here's an example of some of the protocols that we use. There are specific protocols that have been developed to treat different conditions. And on the left you see a protocol that is treating abdominal inflammation, and then the protocol on the right uses, and it's called the odontoids. It's a headband to deliver current, and then you're delivering a current just above each tooth. Each tooth corresponds to a different system in the body. So you can do, this is quite an effective treatment actually for the brain, and even for knee inflammation. When you get to the tooth that corresponds to the knee, you'll find that that's irritated. Or if somebody has low adrenal function, when you get to the bottom teeth where the adrenal area is, you'll find some inflammation there. So it's also beneficial in diagnosing or helping us to determine where some abnormalities are in the body. So in summary, the microcurrent technology is another useful tool in the toolbox to help us treat children with autism and others who have chronic inflammation or hard to treat conditions. It's non-invasive, it's fairly easy to use, and we do see some sometimes very dramatic results in terms of healing and repair. So I thank you again, Taka, and the Holland Biomedical Clinic for hosting this event today.